Hello friends, today we are starting with second lecture of biochemistry, that is property of carbohydrates, and I hope you will love this lecture a lot. So in this lecture we are discussing the properties of monosaccharides, and as we discussed in the last lecture that hexoses are mainly the monosaccharide and they are glucose, they are containing 6 carbon. So I am drawing 6 carbon total, and this 6 carbon sugar glucose contain one of the aldehyde group and 4 secondary alcoholic group. CHOH, they are secondary alcoholic group, and CH2OH is primary alcoholic group. And apart from this, you can see that it is containing 6 carbon. So glucose gives all the tests. First is the reaction of 6 carbon. So when glucose is reacted with, this is glucose, CHO, CHOH whole 4 and CH2OH. When it is reacted with HI in presence of red phosphorus, it gives hexane, normal hexane. This is 6 carbon alkane. And the next one is reaction with showing its alcohol group. Alcohol group, it contains 5 alcoholic group, and out of these 5, 4 are secondary and 1 is primary alcoholic group. So all this group, I am drawing all these groups, CHO, CHOH whole 4 and CH2OH, when this reacts with 5 molecules of acetic anhydride. 4 I have written in place of secondary alcohol and the one with primary alcohol. This forms a compound which is having CHO, CO, CH3. How it is formed? Because CH3, COH is removed in every reaction and ultimately penta-acetyl glucose is formed. And the reaction is with acetic anhydride. So this contains these are the reactions which shows it contains 1 of the primary and 4 of secondary. Now the reaction of primary alcohol group, for this I have to go for oxidation. So when glucose, CHO, CHOH whole 4 and CH2OH, it is the glucose, it reacts with a mild oxidizing agent like bromine water. The aldehyde group only changes to acidic group, that is COH group. So this you are seeing, and this compound is called as gluconic acid. But if it is treated with a strong oxidizing agent like HNO3, which is a strong oxidizing agent, then the aldehyde group as well as primary alcoholic group, that is CH2OH, they both changes to COH, and this oxidation is ultimately forms saccharic acid. So both are oxidized. Now I am showing you the reactions which are important, reaction of aldehyde group. And the aldehyde group reactions are important. Why? Because they form a oxygen test. They are oxygen tests. So CHO, CHOH whole 4 and CH2OH, it is again a structure of glucose. This combines with phenyl hydrogen. This compound is phenyl hydrogen. This reacts and ultimately water is removed from this, and CH double bond and NH and this benzene, and the rest structure remains same. This compound is formed. In presence of excess of phenyl hydrogen, this compound forms oxazone. So the structure of oxazone is CH double bond and NH benzene ring. The next CH and second carbon again utilize the same structure is formed, and the rest 3 carbon of CHOH and CH2OH. This is oxazone. This oxazone formation is important. Why? Because this is a test, and in this test crystals are formed and with different shape, so we can identify different sugars like you can see these are the needle shaped. This is the structure of glucose crystal. The next I am drawing is sunflower shaped structures, and this sunflower shaped structures are maltose crystal, and the last is cotton ball shape. I'm trying to draw it, and this structure is for lactose sugar. So you see different shape, needle shaped. The second one is sunflower shaped, and the last one is cotton ball. So by seeing the different type of crystal under microscope, one can identify which sugar is present, whether glucose, maltose, or lactose. So this is test. That's why this reaction is important. Now moving to inedible. This is next one more test which is given in time formation. You know that in mild alkaline medium, these sugars they form a compound which is called as enediol. Whether they are aldehyde having aldehyde group or ketone group, if both are free, they ultimately form enediol, and this enediol is formed because of tautomerism. Tautomerism is a type of isomerism, and this enediol is highly reactive species. So it is used for the reduction purpose, and sugars are called as reducing agents. Now I'm writing the reaction. This is structure of glucose, CHO, first is aldehyde group, drawing 6 carbon, third carbon containing OH and H, rest are H and OH. This is glucose. Now I'm drawing the keto sugar, that is 6 carbon, again fructose. First and last is CH2OH, and second is a ketone group. Rest structure same as that of glucose. They both in mild alkaline medium, they form enediol. So this is the structure which I am drawing. This is the structure of enediol, and the last one is fructose structure. Now you see why it is called as enediol. The keto sugar, fructose and aldehyde group, en is double bond and 2 of OH group, that's why it's called as enediol. And enediol is highly reactive species, and if it is highly reactive, it is reducing in nature. Now the principle of Benedict's reagent and Benedict test. This Benedict reagent contains three parts. One is sodium carbonate, the next part is copper sulfate, and the last part is sodium citrate. So these parts, the first part sodium carbonate, it provides mild alkaline medium which is required to convert the sugar into enediol. The copper sulfate, it provides copper 2 plus ion, sodium citrate is stabilizing agent. When this Benedict reagent is added to sugar, the sugar plus Benedict reagent, they ultimately gives a red PPT. How this red PPT is formed? That sugar, they are converted to enediol with the help of Benedict solution. It provides copper 2 plus ion. Ultimately, red PPT is formed because of formation of cuprous ion. And this mild alkaline medium comes from sodium carbonate. Benedict solution contains copper 2 plus ion which is ultimately reduced to copper ion, and this is test. If this video helped you to understand biochemistry in an easier way, please like, share and subscribe to my channel.